So, welcome everyone to this month's ReproNim webinar. We are super pleased to be joined today by Tim Errington. Tim is the Director of Research at the Center for Open Science. Tim received his PhD in microbiology, immunology, and cancer biology from the University of Virginia, a master's degree in molecular and cell biology at UC Berkeley, and earned a BS in biology and chemistry at St. Lawrence University. It is always our pleasure to welcome people from the North Country of Northern New York to join us. This may be a first for our webinar series, and thank you for representing New York and the outsides of the city. At the Center for Open Science, Tim conducts and collaborates with researchers aimed to understand the current research process and evaluate initiatives designed to increase reproducibility and openness of scientific research. Included are such large-scale projects as the Reproducibility Project on cancer biology and the DARPA-supported Systematizing Confidence in Open Research and Evidence, the SCORE project, as well as evaluation projects of new initiatives such as open science badges, registered reports, and a novel responsible conduct of research training program. Center for Open Science is a ReproNim service project, and together we have plans to look more closely at linking pre-registration of analysis to the metadata and provenance that the post-execution of such an analysis will generate. Center for Open Science has done fabulous work in looking at the reproducibility of various biological areas. And for us in the neuroimaging community, it's super valuable for us to better understand what is being found in other disciplines as a teacher and a guide to us in terms of the topics that we need to attend to for our community. With this in mind, a recent paper in eLife that Tim led on the topic of reproducibility in cancer biology, challenges for assessing replicability in preclinical cancer biology, caught my attention as something we should pay attention to. So without further ado, I will turn the floor over to Tim, and thank you for joining us.
Great, thanks for that introduction, and thanks for the invitation. And yeah, so today I am going to talk to you guys about these papers that I just published from this project I finished, that I started when I first started at the center. So it's been a long time. Before I do that, let me just first give just disclosures here. You can see that on the screen, just in terms of where our funding comes from, different organizations, philanthropic groups that support it. And I'll come back and highlight that a little bit, just in terms of the project who supported us. All right, for those not familiar with the Center for Open Science, we were founded in 2013. We're based in Charlottesville, Virginia, and we have a broad mission to improve openness, integrity, and reproducibility of research. What I like that is it's agnostic to discipline. So Brian Nozick, our CEO, is a social psychologist by training, and myself, as you heard, is a preclinical cancer biologist by training. And we do these activities through those different arms of infrastructure, developing the tools that you just heard, some policy work, working with journals, and then meta science research, which is what I lead. And so I'm going to talk to you today about just one project in particular, and then I'll end a little bit with a couple slides, just high level view of what's kind of going on in this space. But before I do that, let me kind of hopefully bring you down into where I was when I started this project in 2013, which is the same time when the center started. So in 2013, at that point, there had been a couple papers that came out, a whole bunch, but there's a couple that really kind of caught our eye in terms of kind of suggesting that the literature was a bit too good to be true. So one of those that you might be familiar with from 2005 by John Anides was basically suggesting that with such high positive results in the way that the research was conducted, that he thought that a lot of the published findings just couldn't be true, just based on definition, the way that we were kind of conducting our research. That was done largely by looking, not empirically looking at the data, but looking at the way that we were communicating and sharing our results. So a lot of that is kind of a simulation, you can kind of think of it. The ones that really caught the attention in the preclinical space, especially in cancer biology, were from two pharmaceutical companies, Bayer and Amgen, that had reports, just white papers in 2011 and 12, that basically said that they were having a hard time. They weren't doing this proactively. They were looking kind of back retrospectively of replicating research that was published in the papers that they were kind of using to feed into their clinical pipeline. What was interesting about those, and I still remember when I was a grad student reading these, was they had a strong call to action and a very, I think, still probably controversial statement of saying that there's a concern about the replicability of research, but it lacked the details that one would normally want, right? So it didn't tell us, gave us a number, but it didn't tell us, well, what did they mean by that? What were the papers they were looking at? What did they find? How did they assess replicability, right? Something that's very challenging. What were the barriers they had? Glenn Begley in particular, he elaborated a little bit on this in terms of some of the issues that he found, kind of thinking back to it, but that's more of a qualitative approach, his opinion. So in 2013, we wanted to do this empirically. We wanted to do it one more time and do it in the open. At that same time, Brian Nozick had just started the Center for Open Science. It's just when I started, and he was working on a project in psychology, doing it in the open as well. And so when I started, I modeled the project off of that project, that psychology project. And with this, what I'm showing you here, these papers kind of being the stimulation for it. And just before I go into the project in some more detail, one of the reasons we were doing this is also trying to understand, like, well, is that those papers were of concern, but is this actually something that we should be worried about? And again, I might be speaking to people who are aware of this, but this, is an interesting paper or survey that was done kind of informally through Nature. Moni Baker did it. You guys might be familiar with this, asking that question of, is there a reproducibility crisis? Which I don't really care too much for the word, but I think it's a fair word to use. And a lot of respondents, and this is both grad students and senior professors and across a lot of disciplines, said yes, there is a concern. What I find more interesting, if you've ever looked at this paper, is why you failed. Why were you having a hard time to replicate or reproduce an experiment? And what's interesting here is it's not just somebody else down the hall. It's not just another paper. It's even in your own work. And I think that's, to me, and I'll kind of go through what I discovered in the project, is something I think we should think really hard about, is how do we actually communicate our own findings for our future selves? That's probably the biggest beneficiary for this whole process. Okay, so in 2013, we started this project and we received funding to do this, not from NIH or not from any other large biomedical foundation, but from a new, at the time, philanthropic group called the Laura and John Arnold Foundation, which is now called Arnold Ventures, on that first slide that I was sharing. And they did it because they were a new group and they were looking to invest, and they kind of understood that the investment is only as good as the kind of research that goes into it. And they were concerned about where to invest their funds philanthropically. And they started to realize that part of the way they could do that is to invest in the integrity of research itself. We did this also with our partner Science Exchange, which is an organization out in Palo Alto, California, that essentially is like a marketplace to find researchers, largely in the biomedical space, but also in the clinical and the chemical space. And you basically are able to just suggest, like, these are the types of experiments I want, these are the techniques that I'm looking for, and you can find experts that have those abilities. And they kind of fit two flavors. They're either academic core labs, right, like your microscopy core or some other shared facility at an academic institution, or they could be a contract research organization, right, an organization that pharma uses themselves to contract out experiments. So this meant that we had a network through Science Exchange of technical experts within the specific types of experimental techniques that we were going to look at. And then the last thing is, before I show you some of the details, is we also looked for a publishing partner, because it is hard to publish replication results. And we thought that it would be best to have this on the front end. And so we found a great partner in the open access journal eLife, who agreed to kind of publish everything that we were doing and give it independent peer review. So it's a bit of a checks and balances at the same time. So we worked with them over the years on this project. Okay, so what did we do? So in 2013, the approach we took was to first sample the literature. So we went back 3 years, kind of arbitrary, right? 2010, 11, 12. And we were looking for papers that were making a high impact in preclinical cancer biology. So we did that by not looking at citation rates, but looking at like downloads, Mendeley readers and those types of alt metrics. To find out, well, what's getting the most attention from these 3 years. And we were looking again at that preclinical cancer space. We would look under search terms like cancer or onco. And of course, we'd cut out anything that was like a review or clinical. We were only hunting for the preclinical. We also stayed away from kind of those omics experiments, so genomics, proteomics, and the such, or any high throughput assays or novel techniques or reagents, because we were more interested in the types of papers that, whether they were or not, indicating that I'm doing more confirmatory research and doing it more in those like kind of classic cell biochemical molecular biology techniques and animal studies, which there's plenty of. And the reason we did that is, one, we were looking for that particular space because that matched to the Science Exchange network very nice. But two, it also gave us a bit more of a box to kind of put the sampling strategy around, because the genomics and proteomics communities, they just have different challenges than the ones in those wet labs. When we set out to do the project, we thought we were going to just be looking at outcomes and maybe doing a little bit more of descriptions of what we did along the way. at the end of the project, after 8 years, I realized that the only way to do this was to tease this apart into 2 large papers, and I'll walk you through those, which was, well, what were the challenges of actually trying to test this, of actually trying to do the replications? And then when we actually did that, what did we find? How do you actually tackle that very difficult question of, did it replicate or did it not? One of the approaches that we took, and you can see this kind of in a diagram on the right, this is something Brian and I wrote a couple of years ago, is we really wanted to kind of bring in as much rigor as we could. And one of the ways to do that, besides just making sure you have the right sample size from power calculations and controls, was also to really think through the process. And to us, pre-commitment, what pre-registration essentially is, and registered reports is an extension, we thought that was the most crucial part. So again, this diagram kind of gives you a simplistic view of, if I was to replicate something, I was to decide to do it, which anyone can, right? Because we should always be doing this. If I don't talk to anyone, or I don't have any pre-commitment, and I just kind of go on my way and replicate it, what you end up is a lot, and you see it when it does happen, which is a lot of quarreling. Oh, you should have done something differently. Oh, you designed that study differently. You used the wrong material. And so you end up having a little bit more of a back and forth, and nobody really agreeing on something. What we wanted to do was get way ahead of that and have pre-commitment from the original authors as much as possible, but other experts to say, well, this is exactly what we plan to do. Does everyone agree to that? We believe this is the right way to analyze it. This way, when we look at those outcomes, even if we disagree on what the implications are, we can't really disagree on the design, because we decided that beforehand. We agreed on it beforehand. And so essentially what we're doing is we're confronting our prior self. And we thought that was an important aspect that would help us advance. It's something that they did in the psychology project, and we carry that forward here in the cancer biology project. But we did this through the journal. And I said eLife was our partner for it. So let me give you just kind of a brief view of this in case you're not familiar. If we think of the traditional academic publishing model in this very simplistic form, we develop our ideas, design what we're going to study, our experiments, and collect that data, analyze it, write it up, then shop it around, get it peer reviewed. Hopefully after that, it can get published. Importantly, that peer review, that external kind of step, occurs after we've done all the actual scholarship, after we've done the design, after we've actually collected it. So it's really hard, because a lot of the critique is post hoc, and it's not really helpful for us in terms of designing the best test up front. Not to suggest that we as scientists can't do it, but we always want that input earlier to kind of hold our biases at bay. So a different way of doing this, another format that one can use that we've been advocating for, is registered reports. So for those that aren't familiar with it, it kind of creates a 2-stage review, which is you have your idea, have any pilot data, design your study, and then that's where you get peer review. And then if that passes peer review with the input and revisions, then you can actually just collect your data, analyze according to what you said you do, write your report. And then that second stage is just really not about, oh, is that an exciting result or not, which is kind of how most peer review goes. It's more about adherence to those pre-commitments. So we use registered reports in our project through eLife, largely to increase the credibility of the research, but also to make sure that it got out there openly. All right, so let me give you a diagram. Somebody had done this years ago, which I think I still reuse, kind of outlining the approach that we were taking. So like I said, we had these papers across these years, and with some exclusion criteria. We'd identify kind of key conclusions from that. That's done by us. The way we kind of defined that was we wanted statements that were in the abstract, and then we'd identify experiments that kind of spoke to those claims, again, excluding things that were in the omics space. So for example, if there was a paper that had a huge genomic screen, we wouldn't do any of those genomic experiments, but if it included within it, say, a test in an animal model on some targets to kind of validate their screen, that's what we'd interrogate in the replication, and especially if it was spoken up into the abstract. So we'd identify some experiments. And again, these papers that we had were largely Nature Science Cells. So there's dozens and dozens of experiments. We only did a subset of those. Then we'd start with the paper. We'd go right into the paper and start reading it and making sure not only that we knew the experiments were, but what was actually done, right? What were the results that they found? What was the methodology they used? Trying to understand as much as possible from the paper. And there's challenges there, and I'll get into those. Then we'd contact the original labs, right? We'd always reach out to the original authors, sharing what we had and always asking for input, whether it was in the paper or not, always trying to do a good faith replication. And then again, we'd eventually contract with labs from Science Exchange, and we'd have some back and forth, trying to optimize, well, what are we actually going to do? Because you can never do the exact same thing twice. So we're always going to make changes. We just wanted to make sure that all that was checked. After we had that, then we'd send that information off in step 4 to eLife. That's when we'd have peer review done, not on what we found, but what we were planning to do. So the original authors would be invited back. So we did it informally. eLife would help us do it formally again. There'd also be other experts in the field and a biostatistician as well, who's kind of looking at our protocol and saying, are these the right experiments you should do? And is it articulated well? And is the analysis strategy appropriate? And so there'd be revisions and back and forth there as well. And then only after that did we actually conduct our experiments and then publish those as a replication study. So we always published 2 papers for every single attempt on it. We had the registered report, our whole protocol, and then the replication study, all of our outcomes. Okay, so what did we find? So I'm going to take a step back and walk you through this. I'm going to look at we can do this at the experiment level or the paper level. I tend to do this at the experiment level. It's the one that makes the most sense to me. All right, so when we were first designing this, we had 53 papers that we were interested in, 193 experiments that we had selected. And so the first thing we wanted to do was to get access to the data. That's important because we want to both understand what the effect was that was found, but we also want to make sure that we're powering our experiments appropriately, right? We want to make sure that we have the right sample size. But we had challenges right there off the bat. Only 2% of the experiments had open data, right, that we could actually access at the raw level. Even if we requested, only 60% could share raw data. So a lot of the times all we'd have is the paper. We'd have bar graphs in the paper with error bars, and that was it. So we'd have to estimate from that in order to move on. So a barrier, but we could get over that. Same thing if we were trying to understand what the statistical analyses that were being reported in the paper. One, not a lot of them would report. So it's not common in this field to report statistical analyses. A lot of the times, because it's just a representative image, it's all you've got. It's just a single image. You don't have any other variable data. But the times when we did have it, about a third of the time, 21% of them, we had no idea what the test was. All we'd get is an asterisk, p less than 0.05, and maybe we understood the comparisons and nothing else. So again, we can get over that, but it's a huge barrier to understanding what was actually done. And similarly, code sharing was also a barrier in terms of the analytical strategy. Only 1% actually had their code open so we could look at it. And then again, after request, only 13% actually shared code. And by the way, I'm being very liberal here in the definition of code, which is if you did it, Prism is a pretty common file that had, but even if you just shared your Excel file and you just, did it all in Excel, that counts because I can understand your analytical strategy versus just the asterisks that is typically reported in papers. But again, we can get over this. Just what you're seeing is barriers, and we have to keep making assumptions over and over and over again to understand the paper in order to replicate the paper. But it gets more complicated because eventually what we run into are key reagents that we need in order to actually do the experiment. So by key reagents in this space, it's animal models, mouse models in particular, cell lines, antibodies, plasmids in order to conduct experiments. So when we were looking into these experiments, if we were hunting to see if we could get access to them, or at least understand what they were so we could understand where to get them. And what we realized is that these key reagents, we had to ask 70% of the time for them. That means they weren't openly available in like AdGene for a plasmid, or ATCC for a cell line, or JAX Labs for an animal model, or they weren't even commercially available at Sigma. So the only way to get those was to go back to the authors and find it, or to remake it. Thankfully, 69% were willing to share, so we could move on. And we'll come back to this. Just as a note, and kind of anecdotally, this started to create huge problems within the project. Early on, we ran into one example where the paper we wanted to replicate included an animal model that had a triple transgenic knockout mouse. We went to the lab because it wasn't deposited anywhere, and they told us, no, we're not going to share it with you. You have to remake it yourself. That would have taken us years to remake just the model to try to replicate their experiment, and who knows how much resources just to create the exact same thing again. So we ended up dropping that paper. We couldn't proceed. And those were the types of barriers and the kind of discussions that we had to have as a team, which is how are we going to proceed if we don't get access to these things? Do we replace it? And then if we do, is it a valid replacement? Do we remake it at cost and time? Or do we drop it or attempt some other mechanism to get it? Okay, so on top of that, not just those key reagents, we also had to understand the methodology. And so for anyone who's ever looked at a paper, that they lack. And I think this is something really to think hard about, which is how are we communicating our methods, our protocols in the paper? So like I said, we'd always start in the paper. They were always missing information. So in some ways that's surprising, but to me, it's actually not surprising because that's a really poor way to share. What was more surprising to me was that even though they all needed clarifications, a fifth of the time, they were really important. They were like, we didn't even understand what was going on. Really vital clarifications. And I'll give you an example in a little bit. And that meant that if we didn't get that information, we were about to make huge, huge assumptions in terms of the way that original experiment was done and how we were going to compare it with our replication experiment. And again, we'd always ask the authors, right? And a lot of the times that's what you're supposed to, it's the whole point of corresponding authors. A lot of times that tells you how to get additional information. Still goes on today, right? We'll seek, if you want the data, if you want the code, email the corresponding author. What we found when we were doing these replications is it was very bimodal. We'd have a lot of people that were incredibly helpful. And I'll give you examples of how helpful they were. But we'd similarly have a third that were not helpful at all, had no responses. We'd get, if they did respond, they'd say, I would help you, but you're doing a replication, so I'm not going to help you. And so I think that's, again, something to take pause on, which is how can we change that culture? How do we make it so that it's not, one, we're not as dependent upon it. So how do we, expose, be more transparent? But two, how do we also recognize that replication is something that helps us advance our understanding, not necessarily as a threat. All right, so let me describe what I just told you in a slightly different way. So here's an example of a paper up on the top left here. You see kind of a classic thing. There's a figure, this is a science paper. We've got 4 panels. We got a representative image up there from a cell biology experiment, some bar graphs, some asterisks, right? So that's pretty classic. We see a lot of this, and that's the type of experiment. We just wanted this one experiment. We go to the materials and methods. Here we have to go to the supplement because there's no room in the paper. It's very brief, and usually what it does is it tells you to reference another paper. So we'd follow those references and you'd go down, and next thing you know, you're 5 references in 20, 30 years in the past saying, and you can't even find the methodology. And that's the culture we're in, right? Which is that in order to describe what you did, you go back in the past and there's nothing there. So we would try that. Usually that's what we'd find is nothing. Sometimes you'd find something that was useful. But regardless, we'd always send a Word doc back to the authors, which is a screenshot there on the right on OSF, highlighting, here's what we think you did. Here's our questions. We're not sure what you did in these steps. Can you help us out? Or can you correct us if any of this is wrong? And of course, can you share data, key reagents? So we'd kind of compile that and reach out to the authors and work with them if they were willing to work with us. And so again, what were the types of things we were asking? Well, they were anywhere from relatively mundane to incredibly important, right? So we always wanted to know, say, what antibodies were being used. It's really important, right? Especially when they're something like MYC antibody. Well, there's tons and tons of MYC antibodies. Which one did you use? How did you use it? That's really important because we know that we want to make sure we're using validated reagents and validated protocols as much as possible. What were the sex of the mice, right? A lot of the times we'd have no idea. And so that'd be really important, obviously, to compare experiments and make sure we set up ours right. We wouldn't know key things like how long the treatment period was, right? It'd just say I injected it with a drug and then I sacrificed the animals. You're like, well, how long did you do that? What was the dose? Sometimes we don't even know the dose. We just know that there was drug injected and that was it. We wouldn't know the software, the instruments that were being used to collect it, right? Think of imaging, we would have no idea what they were using. So we'd have a very hard time to say, well, that's great, you have these images, but what did you actually use to collect those images? All right, what was the protocol? How did you define, right? Positive area. Okay, well, there's many ways to define positive area. How did you define it? Because all I see is a bar graph that says positive area. So these were the types of questions we had to really understand what was going on in the original paper so that we know what we were going to do in our replication. And again, data can help a lot, and we'd always be asking for that for every experiment. So the responses we'd get back varied a lot, right? It wasn't always like, oh no, I'm not going to tell you it. It was actually that it wasn't set up for what we were doing, right? The system wasn't set up. Their labs weren't set up. They weren't individually put in the right place, right? And it goes even within the incentive system. When we think about publications, right? We'd get responses, well, yeah, would have had it in the paper, but we had to cut down, right? There's a text restriction, so we didn't have room in the paper. So we lost it. That information's lost. There's a good example of someone that we were kind of trying to sort something out. And eventually he shared with us a prior draft of the manuscript that was submitted to the journal, because when they actually had it accepted, they were told to reduce and cut out figures because there was a figure limit within the paper. And it just so happened we were hunting for something that got cut out. So that's a challenge. It's a challenge to not have that information shared easily. They wouldn't have the information. We definitely had the equivalent of the dog ate my homework. Good example of that is someone who was a postdoc. They did their work. They went off, started their new lab in a different institution. They said, oh, if you want the data, go back to the original lab, right? Just a couple of years out, but they're the ones that have it. I've left. You'd ask that original lab, okay, well, can you share the data with us? And they were like, oh, that was on the laptop they were using, and we wiped it clean when they left. There's no more data. So it's gone, right? So we were like, okay, great. So all we have now is the paper and nothing else. The original data is all lost. And it's a challenge too, because these are not single author papers, right? These are huge collaborative networks. People have moved on, and we would have people that could help and hunt it down, but all that was a challenge for them, right? Because once that paper was published and people left the lab, it's hard to put it back together if it wasn't shared in the first place. Like I said, they don't have the data or the details readily accessible. And that's something that we can definitely come back to in terms of what we can do to improve. All right, here's a little bit of something that comes through it, which is, I focused a lot on the questions that we had and the responses we had. And, one thing that we were very interested in is that kind of very obvious hypothesis of, well, depending on how depending on how much we asked of the authors, so the clarifications needed here on the x-axis, and you can see this by paper experiment view, that's how helpful the authors were, which is the y-axis in both of these. So hypothesis, well, maybe there's a relationship between the two, right? We ask for more, we get less help. There is a modest correlation there that maybe the larger requests, did have some less helpfulness, but by and large, it doesn't explain everything. And it really does give you a good view here, especially if you look at by the paper level, that bimodal response, that authors were either very helpful or they were not very helpful. But still something to think through in terms of like what we were doing in terms of the burden that was placed on our requests. All right. So eventually we were able to keep going. And I think you can probably appreciate and understand why we had to cut back on the number of experiments we were doing, because we had to put resources into other experiments, right? Remaking reagents that weren't shared, right? Or having to try to navigate all of the time and effort it took to even understand what was done originally. So eventually that number got dwindled down to 87 experiments. And we still had some challenges, and some that were not ideally expected. So back to those reagents shared, we had a lot of people that were willing, 70% that were willing to share with us, but we didn't actually get them all. There's a good example of one where they were willing to share the plasmids. They ended up not sharing the plasmids. And so we had to remake them, right? So all of a sudden you're like, well, shoot, here's something we didn't anticipate. So now we have to spend more time and more cost on remaking this reagent that somebody said that they were going to share, and now they're not going to. Sometimes, usually we'd remake it because that was usually the path of least resistance when we could do it. Sometimes we would, unlike that prior example I gave you of the transgenic knockout mouse. We did have some cases where we actually went back and said, well, will you enforce your policy? There's one time we had that, the lab was not willing to share a whole bunch of reagents with us. We tried to do the MTA. We actually got responses back from their material transfers office that was always like, yeah, that they won't sign off because you're doing a replication. And so we were like, oh my gosh, what are we going to do? We're going to have to either drop this paper or, at this stage, they said that they were going to share, and now they're not. So we went back to the journal and said, will you enforce your policy? And they did. And so the authors shared reluctantly, I'm sure. And it's not really what we wanted to do, because now, that was the way to let us proceed. We did it near the end because we thought that it was better to do that and proceed than to sit there and kind of go back to square one on more experiments. But it was definitely a challenge, more of a cultural challenge as well throughout the entire project. And then when we actually did some of these experiments, what's amazing is that it did work. We were able to articulate exactly what to do and conduct the experiment, and the models and the experimentation would work fine. Whether the results were the same is a different question, but the controls worked. And that was the part that we were really paying attention to in terms of execution. But sometimes they wouldn't. Sometimes we'd have a system, it was described, the original authors would be very helpful, and it just wouldn't behave the same way. Maybe the animal model, we had one where the animal model looking at tumor growth in vivo. So we would do in vivo imaging. And then only after we saw the tumor actually take presence, it was a blood cancer that we injected into the mice, then we'd randomize to treatment and then keep tracking the mice. We had an issue there. We couldn't actually detect the disease in the animal visually. We couldn't actually see these cells growing. And then we kept doing it over and over. And then eventually the animals did get sick from disease. So that meant the model wasn't behaving. The timeline was all messed up. So we had to go back to the authors, and they gave us a whole bunch of different optimization strategies. And we spent time and eventually got it to work with a very, very different strategy from the original one, because we had to modify what we were doing beforehand. And really, to me, what that speaks is that we need to have a lot more attention to the control models that we use, not just the outcomes, right? Treatment versus control. Because we ran into that a number of times, not always, but we'd run into those problems. And again, that means we're spending more time trying to figure it out before we can actually proceed and understand what we found in the experiment. And that's what we found. We were able to complete them when we had to do them, much of the time, but eventually we just ran into a strategy where it was not worth pursuing farther. We were spending a lot of resources trying to continually optimize it. And at some point we just stopped. We kind of ran out of time and resources to continue doing that. It's kind of this question of how far down do you go before you just kind of give up and say it's not worth the return on investment. And we had to struggle with that constantly throughout the project. So a lot of unknowns as a result of it. And when we look back at the types of modifications that were needed and whether we were able to implement those, again, by paper or experiment, we've got the needed on the X and whether we were able to actually implement them on the Y. We see there's no correlation between the two. A lot of the experiments didn't need any, that's that box in the top left. but still something that we were curious about, but we don't really see a relationship between our ability to implement modifications depending on how difficult they were. Okay, so I have not talked to you at all about what we found. I talked to you entirely about what we were trying to do and the challenges that kind of came along the ride. So I'm going to summarize here just a bit before we do get to that second question, which is, well, how many replicated? Well, the reason we had a hard time even testing if something could replicate was a lack of transparency and access to methodology and sharing data and reagents. That simple step of sharing and transparency was a huge barrier to us. That also meant that we were quite dependent upon communication with the original authors. And if we didn't have that again added an extra burden on us, both for us needing to make up the lack of information, but also on the original authors, because that's a lot of ask of them to try to fill in those details that weren't there in the original paper. And again, we had unexpected challenges, like I just mentioned. And really what it gets to is just a lot of inflation of cost, time, and uncertainty by everything that's occurring above it. So a lot, and that's really, to me, it's not even just trying to test, it's trying to understand also what the original paper is. What was the original finding? How was that? How did they do that? A lot of our effort was just trying to understand the original paper, not necessarily even conducting the experiment. Okay, so now let's shift. So we found stuff. We definitely had experiments that worked. So what did we find, right? We had 50 experiments that we actually were able to get effects that we were able to measure. And so now we got to tackle the more challenging question of what does it mean to replicate? What does it mean to get a similar finding or not? And so what we did is we looked at a bunch of different ways to do this. Here I'm showing you kind of five common ones that are used, each of which have strengths, and many of them have weaknesses as well, including the first one. And what we see here on the diagram on the right is just an example. It's just kind of very simple for if I'm not making myself clear of how one can envision it replicating or not replicating. So one is if it's the same direction and statistically significant, that's pretty straightforward, right? If the original has the asterisk, does the replication have the asterisk, p less than 0.05? Which is not a great way of doing it, right? But it's one way to measure. It's a pretty common one for all its flaws. Another one is to use confidence intervals, right? It's the original inside the replication confidence interval and vice versa, is the replication in the original confidence interval. So this is good. This starts to take into some of the uncertainty of those measurements, right? Effect sizes are important, but the confidence that you have, the uncertainty you have is important, and that helps us too with the variability and sample sizes between the two. So we looked at those. Those are common ways to do it both ways. We also would use prediction intervals. It's another way to look at it. Again, has a lot of flaws, has a huger, it rewards smaller sample sizes. So there's a lot of challenge in including this in replications, but it has been used once before. So we included it as another measure. And then we said, well, what if we take the two together, the original and the replication, and do a meta-analysis? Is that statistically significant in the same direction? If it is, then that could be another measure of success. Of trying to assess replicability, because now you're using the total evidence that we have between the two papers or the two findings. All right, what did we find from these 5 different approaches? This is just one way to look at all this data. Well, it depends what you want, right? These are all dichotomous, right? You can say yay or nay to each one, as I just kind of went through myself. And you hit huge, depending on how liberal the approach is, you can have anywhere from 25%, which is actually what Baer reported in that white paper I described way earlier ago in 2011. But you can also see 63% if you use a meta-analysis combining the two. So it's very variable. And this is kind of what we saw in the psychology project as well, that if you look at different measures, you get high variability between the different ones. So we asked a different question, which is, okay, well, each of these have pros and cons. What if it doesn't matter? What if you just say, all right, I have these 5 criteria, all of which are used and have been used, and we use them. We're curious to know if you succeed more often than you fail, right? So do you succeed on 3 or more, or do you fail on 3 or more of those criteria? And we looked at that and our total results, and we found 46% succeeded on most criteria, while 54% failed on most. So if you had to ask me to give you a single number, now I'm happy to do that after 8 years of work, which is, yeah, I think we only succeeded 46% of the time if we use this as a measure of it. As a note, we also, when we were doing these replications, we didn't just include positive results. There's plenty of those, right? Positive result being, oh, I give an intervention and I'm looking for the difference between my treatment and control. And I'm trying to say there's some efficacy. There's either tumor decrease or increase in survival of the animal, for example. We also would include key null results, right? So just as important as it is to look for efficacy, you'd have related experiments trying to make sure that there was no downstream detrimental effect, say on the heart. If you're looking at giving an animal a drug, you want to check that. So there were sometimes we'd include null results. And what we find when you kind of compare the positive and the original null results, recognizing the sample sizes are quite different between the two, the null results succeeded more often than the positive did. And so that was an interesting finding as well. But there's another way, and there's something I'm more interested in, which is not really those dichotomous measures, but actually trying to think about the actual effect size. And I think especially in the preclinical spaces, you think of these as being the evidence that eventually hopefully builds up to the rationale whether we should go to a first in human trial with any of these approaches is that size of the effect. And so what we have here on A is all of the effects between the original and the replication. B is just to zoom in for effects that are smaller. These are on the SMD, the standard mean difference scale. And what you have here is on the x-axis, the original effect size. Everything's positive by definition. We made everything from 0 up. So 0 meaning there's no relationship. And then on the y-axis there is the replication effect size. And you can see here, the colors are telling you whether the replication was statistically significant based on that magical 0.05. And you can get the distribution curves of the original and the replication. What you see is that the replications were 85% smaller on average, right? That diagonal line that goes from basically through 0, not -5 up to the 25. If everything was perfect between the replication and the original, it'd be a perfect diagonal line. What you see is everything's below that diagonal line, suggesting there's a big regression to the mean that's occurring in our replications. And 85% is pretty substantial, right? It's saying if I had, a difference of 100 in the original finding, we're only seeing a difference of 15. That's a huge difference when we think about advancing our knowledge and moving it up towards translational potential. And we can see this another way if we want to not look at that graph. If we just look at, say, the effect size. Again, I'm just going to look at the smaller ones, less than 5. If we either look at all the data, the original positive or the original null data, you see kind of a similar trend, right? The original, everything's positive by definition. Here we're seeing the distribution of those effect sizes in the original. And in the replication, it's way closer to 0, right? That huge drop off that I was just talking about, 85% decrease. And if you like your p-values, that's just as striking there, right? With all the flaws of a p-value. So here we have the original findings. Again, the vast majority of those are sitting below 0.05 by definition, especially in the positive. Positive, by the way, was defined by the way the original authors determined it, not by the statistical cutoff. And then when you look at the replications, they're way above that, right? We're seeing a much broader distribution of p-values. And again, this is eerily similar to the psychology ones, which is kind of, for me, very eye-opening to think about the challenges that we encountered here that were quite different, but the results being quite unsettling and similar. And then kind of lastly in here, particularly because of the space, is we also were interested in comparing animal experiments versus non-animal experiments. Those non-animal would be just something that's in cell culture or some other type of in vitro model. And what stuck out to us when we were first looking at this was if we looked at those dichotomous indicators, like whether they were statistically significant in the same direction, we saw a big striking difference, right? Animals were 12% significant in the same direction compared to 54% of non-animal. And we were like, wow, something's going on here. But when you looked at the decrease in the effect sizes, they're roughly similar, right? 84%, 78%. So that's not striking. What we really think might be going on here, and you can kind of see this from this graph, is that those animal experiments in the original tend to be smaller in the first place. They tend to cluster closer to that 0-0 axis. So we think actually there might be something unique about the animal experiments. They're much more complex, both in terms of the biology that's going on, as well as the factors that you can have influencing it. But the effects, I think, probably by definition, because of those, they're much smaller. And so when you have smaller effects, especially with small sample sizes, the likelihood of them replicating is lower. And we think that might be actually what's going on here. Is it's not animal, non-animal. It's actually just the size of the original effect. But more to be determined here for sure. All right, so let me give you some conclusions just from what I showed you, right? So by and large, the effects were weaker than the original. And you can, depending on how liberal the test you wanted to use, that success, right, can vary. You can have it pretty low, right, 25% or up higher, 63. Like I said, we used an approach to say we don't really care which one. We want to know if you succeed more often than not on 5 different criteria commonly used. Interestingly, we found those positive results were half as likely to replicate as the null. And those, like I said, what I just finished is the animal, non-animal had similar magnitudes, but we think it's got something to do with those effect size in terms of it actually being something in terms of it being a striking difference between the two types of experiments being done. And really, just like I told you, the last section is there's room for improvement. And what I mean by that is it's not, I don't think our jobs per se to try to make replicability be 100%. This would be a fine discussion point. To me, it's more about, we've done a lot of amazing work considering our replication percent is this low. So imagine what we can do if we can increase this and move this, right? We're making huge advances despite the challenges that I just showed you here. Despite the replicability rate maybe not being ideal, despite, effect sizes going from, 100 down to 15 in the replication. So I think the possibilities of us advancing our knowledge there's a lot of room to go up, because that's probably only going to help us advance, our knowledge and our ability to kind of help humanity. All right, that's where I'm going to end with those papers. These are the two things we wrote up. I kind of give you just a view, but there's other ways to look at this data too. There's one that we were interested in and ended up not doing, which is subjective assessment. Experts, right? We as scientists do that. It's not whether the p-value is less than 0.05. It's how I interpret that p-value, right? It's not if the effect size is, decreased by 85%. It's how I interpret that. Is that still meaningful? So we didn't do that. It's still something I'd like to explore more. But thankfully for us, within that editorial process at eLife, it was kind of occurring the entire time. The editors would be assessing what they thought as experts within the field of whether they thought it would be positive if it replicated in blue, whether they thought it was negative, that darker red, which is it did not replicate, whether it was mixed. Yeah, some of the experiments replicated, some of them didn't, which is that, kind of lighter red, or that they just felt like they couldn't interpret it, that gray, uninterpretable. So this was Jocelyn Kaiser at Science magazine was covering our project. She updated this. I helped her a little bit on this when she was covering our last two papers. And I think this is a nice way to assess that. She's obviously doing this based on the citations of the original papers. But what's interesting to me more is that, again, depending on how you just choose to see your glass half full or half empty, if we look at just positive, the assessment saying these are the ones that replicated, we're not terribly far off from that 46% I was just showing you, right? That there's definitely room for improvement, even from the subjective assessment side. So something else to think about in terms of how we do this. And the reason I think that's important is, well, if we say something didn't replicate, but fails to replicate, right? It's really the right word. We have to think about what that means, right? What does that mean if I fail to replicate an original finding? Well, it could be that the original was false positive. I think sometimes our mind goes right there. Just as equally, our replication could be a false negative. Now, we tried our best not to do that. That was part of the reason we put something like pre-commitment in there, was to try to minimize that, but not to suggest that it couldn't be there. There's another possibility that I think is probably the most likely, which is they're both true. The original and the replication are both telling us something. And what we're unable to realize is that there's something that we thought wasn't important, but that is really important in understanding whether that effect is going to exist or not, whether I can actually replicate that effect. And so this is a bit of a complex slide. Brian and I wrote this. It's a PLOS biology paper, and at the end of these slides, I have references. So for those that want to, I recommend, I'd be interested to hear your thoughts on reading this. But we were trying to think about how replication and generalizability by extension fit into our advancement of knowledge and our advancement of maturing theories. And I think it fits into what I was just saying. So if we think about any replication, or really any test, there's a huge theoretical space that our test could be applying for, right? There's a lot of different ways, like I was just saying, to conduct it. We can never do the same experiment twice. There's always something that can change based on the samples we're using, the time we're doing it, the conditions, and so on. And because there is this variability and we cannot test every one, what we're doing is we're testing a subset and then we're drawing inference around it. And as we do replications, so we're basically trying to say, well, we have a theoretical reason to believe that this should replicate based on the way we've designed our experiment, right? That's pre-commitment, pre-registration. And if it does replicate, say on the left side of here on the right panel, it increases our confidence, and it increases potentially the scope of that replicable space and potentially that generalizable space. It lets us understand, oh, okay, well, maybe this is work. This is something that's holding true. And the reason why is because it's not that we know the exact conditions, it's that many of those conditions that we thought didn't matter don't seem to matter. Right? That all the differences that we have between the original and the replication, known and unknown, didn't influence it. We were still able to get the same results. So that's really good. That means we have a better understanding of the space. And again, that can go back and forth whether you succeed or not on a replication. But if you have persistent failure, if you're like, no, I can't get the same result. What we're doing is we're narrowing the space that maybe at some point, if I try hard enough, I can figure out how to replicate it, but it might be under such small conditions that it's not applicable anymore, right? That the utility of it has decreased. And it could be that maybe I can never do it again. And in that case, I don't actually even understand how I found the original finding. And in many cases, that's what we're trying to do in research. And I think that's why replication is so vital and why, transparency and pre-commitment is so important to advancing that. All right, so I'll end just with a couple slides to see we're at time. I love this quote from the Wave Lab, Bucket and Donahoe back in '95 there at Stanford. This is from a computational standpoint. They were doing software development. And so I just in red switched out what they were writing because I think it applies to everything that we're doing today, right? That the article is not, that's the postcard, right? That's not actually the scholarship itself. That the actual scholarship, the work that we do, that we should be communicating with each other is everything that's behind it. It's those materials, the data, the protocol, the code, everything that I just talked about that I had a challenge getting, that's the scholarship. That tells us actually how to recreate something. And that's what we should be incentivizing and working more towards sharing. And so what can we do that? So as an individual researcher, myself included, right? Well, I can incorporate more of these open science practices, right? I can share data, code, materials, digital and physical. NIH, for example, is working on that, doing it not just for those big omics repositories, but doing it for more generalist repositories. So we're thankful at COS that the Open Science Framework is part of that cooperative project that's going on by NIH. Like I said, those physical reagents, we should share those, right? I gave you examples, Addgene, there's a lot of these, ATCC, a lot of groups that do that. And what it does is it helps us not just replicate, but reuse those reagents for other experiments. Otherwise, we're just going to be making them over again and wasting kind of our time and effort there. Definitely share those protocols. I do that myself, right? And each of these papers, I still write the blurb that's a summary of the materials and methods that we did for the publication, but we always link out to the actual protocol. All right, we host those on things like OSF or protocols.io. If you actually want to understand what we did, share the protocols the same way we share code on GitHub. And I think pre-registration registry reports is a great tool that can help us, especially as we start to really think towards testing. Do we understand the conditions that are necessary? If I want to do confirmatory studies, this is a great tool to do that because it essentially holds yourself accountable to what you believe is necessary. And then you've got to get confronted on those results, which is if it doesn't turn out the right way and you're like, oh, I'm missing something, you're confronting your past self. And that's a great way to, I think, proceed. And basically what it means is it's more than just data. Data is important, but that data iceberg, there's so much more below it that we should be sharing and thinking about along our journey. And it's not just you as a researcher, right? You need to make sure that that community around us is also practicing those and that those are synced up. So institutional policies, right? Wherever your institution is, how can you align those, right? So that the hiring, tenure, promotion are also aligned with these open science practices. The National Academy of Science, Engineering, and Math has been working on this in their roundtable, trying to get institutions to shift those hiring criteria and those promotion criteria to align more with, not where you're publishing, but these open science practices of sharing outputs. Same thing goes for journal policies. Same thing goes for assessment of researchers, right? It's not about the citation impact you have, although that's the way we do it now. We should shift those and think about the metrics that we do and how are we aligning those with everything that we want to do as researchers. And that helps a lot too, especially early career researchers, of making sure that it's not just saying you should do this, but we support the training of those practices to make sure that we're able to incorporate them. And again, I think that there's a lot of great things that are going on. Just giving an example there of Open Science MOOC is a good one. But really the point here is that we're a part of a social system, right? We as researchers are part of the entire ecosystem, and all these different actors are at play and they can help influence it. So I'll stop there. You guys recorded this and these slides are available there, and I give a bunch of links. And then just the last slide I've got here is also a bunch of references for all the different papers that I was talking about. And otherwise, thank you. And I think I'll take any questions, I think.
Thank you so much. That's fabulous. So much of the ideas that you're presenting are the types of things that we've found important, but sometimes I feel like we're saying it without, sort of like where you started, without an awful lot of evidence. And again, it's just so great to see some of that evidence even in other fields as we push to sort of improve a lot of these same features. I love your referencing to of the important people being actually just us, both in the future and in the past. I think that's hugely important to keep in mind. And I also love the idea that when you go back into the past, there's often nothing there. And again, that's just a type of practice that we've got to get out of the habit of doing and into new habits. I'll turn the floor open to other questions that are out there. JB.
That was great. Thanks a lot, Tim. The exact same sort of thing where I really try to present to my students on the brain imaging aspect, and this is like such a great summary of all the problems. And yeah, one thing that I missed, I think, a bit in your talk, I was wondering, obviously, as you said, like the replication, the null non-replication aspects could very well be lack of power. And I may have missed, did you actually try to set a power aspect to your replication? Because I didn't see that either in the figure, like the original, reproducibility of, Nosec, had that power, like, size to show us, whether, like, if you don't replicate, that doesn't have to mean anything, like, it just like, you may not have the power. And knowing also a subsequent question is not knowing that you have a reduced size effect of whatever, like 80% or, even 50%, how do you actually set power, for future replication if you have a bit of that pre-knowledge that, to some extent we knew a little bit just for the aspect of file drawer effect and the winner curse effect. So just for those aspects, we know already that we are, biased with power, like with effect size. So yeah, so
yeah, there's a lot there. Yeah, so I can start with the first one. Yeah, so sorry I missed that. And it's not in the graph. You're right in the sense that we didn't add that in terms of what we had. Everything we had was 80% power or higher for all of our applications. Okay. That was like the benchmark that we'd have. And when we
sorry, if I can interrupt,
go ahead.
Just like if that's the case, your results are even stronger because because that means like 80% on overestimated effect size is probably like a 95 or whatever is the number. So the null effect is actually starting to be interpretable.
Yeah, right. Right. No, absolutely right. So that was what we set with. It was arbitrary again, could have done different ways, lots of back and forth with peer reviewers over that, I'm sure. But yeah, everything's 80% higher, some much higher than that. I'm showing you where you can apply the statistics. What I didn't get into this graph, and we have it in our paper, which is there are a lot of experiments we had also where all you're left with is the replication, right? If I have an original image, that's it, representative image, we still would tackle it. We just had to do different approaches in sample size. So some of it is even broken and lost in that context. But the graph I was showing is that quantitative approach that we could take, which was the majority of what our experiments were. So yeah, that's what we had there. That helps a lot. Your second question was just trying to say about the you were trying to get at like what other factors to take into place when one does power calculations. Is that what you're getting at?
It just like it strikes me that if I'm trying to get to do replication, we'll try to like look at the original effect sizes, and that original effect size is probably overestimated. So,
what do you do? Yeah, there's a lot of different approaches. We didn't take them here. The SCORE project that you heard from before, Systemizing Confidence in Open Research Evidence, that's in the social behavioral space. There we're doing different approaches. There's some great papers I can send to you guys to reference. Small telescopes is a good approach to take this, which is you can do different ways. The ones that we're doing in SCORE, just to give you an example, is we take the effect size, but we actually drop down and say, all right, well, we're gonna have say 80 or 90% power to detect 50% of the effect size. And we're also doing it where we do different stages. So it's kind of like clinical trials, right? We say, all right, well, up front, we'll do this. We'll do the experiment with the sample size to detect, and I think we do 90% at 50% of the effect size. If we do not detect a statistically significant effect, then we'll increase our sample size higher to try to detect it at 75% of the effect size. But we do that a priori because that's the key, right? If I don't do that a priori, then I'm chasing around for p-values. So there's a lot of different approaches. The small telescopes is another one to think about it, which is you start to look at the distribution and you say, well, because exactly what you're saying, can I just estimate that maybe the actual effect size is a third? Mine would suggest it's even smaller, which is sad, right? It's sad to be like, oh my gosh, should we be doing a 1/10 of the effect size? Is that what we should be anchoring on? But there are other approaches you can take. I'm curious to know which one of those are the right setting. It's not clear to me which ones are, but there are a lot of different approaches, and that's probably the way to go about it, which is you're absolutely right, the original effect size that's being reported, because we know it's going to have a big tilt from selective reporting and publication bias. We should probably be taking something much less. There's a golden rule, and I think the survey community for like social psychology, where they do simple math, which is do your sample size 2.5 times, and that'll get you pretty close. That's another way to think about it. And we did, like in ours, our sample sizes were not 2.5 times as large. That was much larger. But ours were, I think, about 50% larger than the original on average as well. So it doesn't help you because I think it's nuanced to what you have. But I do think it's an important one to consider. And, the more that we can understand the variability of those, models, the better off we'll be with our models and figuring out the sample size. I find that probably the biggest strength that we can have as a community, which is how well do we understand our models and the variability there, because that's probably the biggest factor in determining the power and the sample sizes we should have, right? If I have small effects and highly variable biological variability, I got to really think hard about my sample size because I'm going to miss a lot versus if I have, very tight variability.
I just realized that I said the exact opposite I wanted to say, like the null effects are actually less interpretable, but anyways. Sorry, much time.
There's a question I see in the chat too. I can answer that quick. It's a great question. It was about literature reports and peer review. I don't have an answer to it. I just have, I can give you some thoughts on what we're planning to do to try to get at this. So it is very fair, right? Absolutely right. Papers are not going away. Actually, that's why I find registered reports very exciting, because it can do two things at the same time. It can give us different practices, but it gives us the currency, the same currency, publication, because it's still there. So anytime I think we can win where we're using the currency of publication, that probably is going to have the highest chance of success, right? The worst thing I can do is say, why don't you pre-register, but nobody's going to reward it. That's not going to work well, right? So I say you should do it, but not at cost of your career. But you're right in the sense that already in peer review, and especially here, we're essentially worrying about taxing the peer reviewer, right? There's two sides, taxing the peer reviewer, and I don't know if that's where you're getting, but there's also, I as a researcher, am I slower now as a result of it? It's an open question. I don't know it. Anecdotally, we're not sure, and the reason why is we're not sure we're looking at the right measure here. What I mean by that is, if the idea of registry reports is to help strengthen the type of experiment you're going to do before you do it, I don't know what that looks like outside of it, right? Like, does one researcher just try a whole bunch of methods and then they just publish what's there, which we believe is going on? Well, is that actually, do I need to another way to say this, am I looking at peer review as the wrong measure? I should be looking at the length of it takes to get an experiment done. And those are very different between the two, right? Which is, one, you're kind of doing it idiosyncratically, and then you do peer review one time, and then you either get published or you don't. The other one is you're shortening that. You're thinking more about what I should do, some pilots, then you're putting peer review there that has more time. But the idea is that hopefully I'm spending that time and getting a result that I want, no matter what the outcome is. I say all that in the sense that we don't know the answer, but we're about to launch later this year a randomized controlled trial to test registered reports in kind of the context of a revised resubmit at journals. We've got about 4 or 5 dozen journals that are open to this. And the idea is that for the journals that are willing to do this and the researchers who are willing to consent to be a part of this trial, at their revised resubmit, if they have a new experiment that'll be asked or a brand new analysis, they'll get randomized into status quo or registered report, which will essentially mirror, it's not the perfect registered report, but it mirrors this process. Our main measure is to look at the bias that occurs, right? What do the outcomes look like and how are those experiments designed? But there's other measures that we're interested in looking at, which is exactly what you're getting at, which is what's the burden on the researcher and the reviewer in terms of that review time? That's something else that we're going to look at, because another way to say that is, I think you're calling out the right risk, which is this is a great idea, but there could be risk to it. And so what does that risk look like? And so we plan on seeing if we can measure some of that. It'll take 5 years to do this trial, so you have to wait for that. But we think that's the most rigorous way of doing it, but it's a very good question. Oh, and you want your slides, I'll give it to you.
And is there any savings in reviewer time, in the sense that, is that sort of total review time potentially similar, but just broken up into 2 halves? So is it really taking the reviewer twice as long? Yeah. Again, it's unknown, but do you have to be so rigorous on reviewing the methods in that second phase?
Right.
No, you're just checking, especially if we have ways to kind of document that you did exactly what you said a little bit more thoroughly, and then you don't, well, whatever. You have to trust the researcher to some extent too.
Yeah, you're absolutely right. And again, all we've got is anecdotal here. So, and it's going to vary by editor, but we see some editors that they don't just do it themselves, but sometimes they'll just ask like one reviewer or two reviewers. They'll decrease the burden, because you're absolutely right. It's a different method, because now you're trying to sit there and you're kind of asking two different questions, which is make sure that it matches. And really what you're trying to say to me, and I've experienced it as an author, which is disclose your deviations. And really what you want is a culture of disclose them before you do them too. Like just make it be more open and collaborative. There's tools we can do that a little bit faster. The other one is just presentation, which is, I think, the right place for peer review, which is, are you calibrating your discussion the right way? Are you interpreting it in a valid manner? So I've seen Chris Chambers, I think you may know that name, right? He does a lot of this. I think he does a pretty efficient approach where he's trying to decrease that burden, but he's also very experienced. So he's been trying to work with more and more editors to try to help them say, how do we decrease that burden for everyone, but still maintain the integrity of the process?
Great.
Good question.
And thank you to Qing for adding that question into the chat. If there's other questions out there, I guess the one comment that I'll make, you made the statement, which I'm sure is true, huge advances in cancer biology are happening despite all of this horror show that's behind the scenes in the publication. I'm going to conjecture that the neuroimaging of psychiatric conditions, or the genetics of psychiatric conditions, can't quite hang on to that same huge advances are occurring despite all of this horror in the sort of neuroimaging community. In that again, it's very hard to point to huge advances in treatment in schizophrenia or addiction or autism or things like that over the past 30 years of huge advances. So I think, again, my conjecture here is that, like you were saying, we're really working in super small effect sizes under super huge covariates that are larger than the effect sizes that we're looking for. So it's even exacerbates this more in that domain. So we have to be doubly careful of this as we get to even more subtle biology than the cancer side of things. I don't know if you have any reaction to that statement or agreement or disagreement.
Yeah, no. So yeah, that's fair. I think my reaction to that, and I say this, I know it from my own background when I say it in the preclinical cancer space, which is we make these advances, but it doesn't mean that everywhere that advance is being made, right? So let me see if I can phrase it a different way. There's a ridiculous amount of research going on that a lot of it gets pushed faster than it should, and it fails in the wrong place. So oncology in clinical trials at phase 2 is horrible. The success rate is super tiny. It's one of the worst. And so that means to me, we're shoving so much into that pipeline. And I guarantee you, can't guarantee you, I'd love to study it, to be honest with you, today I can guarantee you. But my high thought process is that it is failing earlier. We just don't see it. We could be weeding that out. We're shoving way too much and we're failing way too late in the pipeline at high cost. And that means what we're doing is we're actually not able to do what we want to do, which is be able to divert the resources into the right places, advance the right place in the right time. This reminds me of a talk I went to years ago where I was looking at the whole drug discovery pipeline. You can measure it really well, and especially in the oncology space, they do it. You can see these slides, right, where you're like, here's how many targets you need. I can tell you the success rate of your screen, the success rate all the way through your in vitro, your in vivo, your early stage 1, 2, 3, 4, and they're like, so they accept all the fail rates, and they're like, so the only way to get more is put more in, right? So I think that's what we do now. Put more into the system, more resources, and you'll eventually get more out on the back end, and you accept everything as status quo. And I think that's what I'm trying to say is we've been operating all of science, especially in the preclinical spaces, that's not changeable. And I disagree with that. I'm saying, okay, well, if you accept that, now let's imagine if we could change those rates. Let's imagine if we say, instead of failing at, succeeding at like 5-10% at phase 2, what if we made that 15-20%, all by investing earlier in the pipeline? I think that's, to me, would be really, game changing in it. And I imagine that would translate into other fields as well. Maybe that's what you're trying to get at as well, which is, okay, maybe we haven't made advances, but whether it's in the clinic or not is actually besides the point. It's more about like, where are you failing earlier and often enough to be able to readjust yourself real quick, or are you just chasing every single lead and that's it? That's just the way we work, right? Is push everything out regardless of whether it's kind of stood the test of time.
Felix, I see a hand raised. Welcome to the discussion.
Thanks. So first, very impressive reproducibility study, really, really cool. One question I have, as it's now 8 or almost 10 years ago, the papers that were put out, is there any relationship to what you just were alluding to, success rate? We know it's pretty low, but is there any correlation between replication of the effect, or if there's a way to assess this, and actual, let's say, success or apparent success?
Yeah, it's a great question, one that I'm interested in. We've been asked this a number of times. I've done some investigation into it. Sadly, there's two big caveats to it. Doesn't mean that it's not worth pursuing. One is it's still too early. So right now I can list a ton of these papers where they're still in early phase trials, or the companies have spun up. So I know that these papers were part of it, right? Because you can trace it. It's really easy. We just don't know the outcomes yet. So that's step one, which is unfortunately you have to wait, which is to me all the more reason to fail early and often, not later, because we're still waiting, like you said, 10 years out. The second one is it starts to get harder and harder to do it. So what I mean by that is there's one of these papers that was looking at a novel drug. The paper we were looking at it in a breast cancer model. All right, so when we did it, we actually did not see at all the same result. We actually saw some really interesting thing with the model. And so that's one where, it did not replicate we still don't know why. I actually truly say it didn't replicate. The editors actually would call that uninterpretable, which I disagree with, but that's why this is fun to do, I think. So you can track that. I still don't know what's happening in the clinic because there's trials going on with the iteration of the drug that has now made its way into the clinic. The other thing that's happened is now it's moving away from that model, right? And actually they tested the breast cancer model in the clinic and they stopped it. And now they're moving to other models. And so now I'm left with, okay, well, why did you stop it? Is there something and is it maybe the problem was not that the therapeutic approach is wrong in cancer at large? It's that the narrow approach, the paper that you presented, that doesn't quite hold out the way you thought it was, and that actually you needed to, and maybe you did over time. Maybe that's why it's taking you so long. Is you slowly realized, no, it's not going to work in a type of breast cancer model that we're looking at. We need to go somewhere else. We need to go to. I think they're now going to blood-borne cancers. They've completely shifted it. So at our level, we can't tease apart the two things, right? We're just trying to replicate the experiment, but it's a great question. And one that I think, sadly, I still have to wait a little longer, but I'd love to interrogate and actually see if I can figure out how far it went. And if we can start to tease it apart. So I think that's exactly the thing to do. But good question. Sorry, I don't know if that made sense, but yeah, I don't have an answer. I kind of wish I did though.
No, thanks.
Great. Steve, you want to jump in here?
Yeah, I loved your talk. I really enjoy this kind of this field and digging into this sort of forensic statistics in work, which is a lot of what I do. I was reminded, not so much that I have a question, just a commentary that it may be changing slightly. I came across a meta-analysis the other day that included whether or not a study was pre-registered as a factor in their model. And what they found is that studies that were pre-registered exhibited lower effect sizes than studies that were not pre-registered. Now, this is in the field of homeopathy, but still, it was a meta-analysis that looked at pre-registration as a factor. So it could be a sign that things are slowly changing in a direction towards this reproducibility and openness that we all want.
That's really cool. Yeah. Can you send that to me? I didn't catch that one. You got me very interested in this. So I love this stuff. I think it's so fascinating.
Sure. Let me see if I can find it.
Yeah. If not, maybe send it to like David or Julie. They can send it to me or my email address. But yeah, I'd love to. I can look it up too. But I think but that's a good thing to think about. Like I agree with your assessment as well. Like, and I think you see it a lot around data too. You're starting to see more and more about sharing practices slowly permeating more and more in there. I think we've got to get past it. I think that's cool that you see it in pre-registration. I think to me, that's like such untapped potential to really think through and transform. So I agree. That's really cool. Yeah, share that. Oh, nice. Thank you.
Just put it in the chat. Yeah.
Now is the rationale for why that decrease might happen is, less sort of harking or, sort of over searching statistical spaces until you find something that you can get a bigger effect on, or
who knows?
All of the above.
Yeah, there was one earlier that was looking at like large clinical trials. This is an older paper now, and you've got, especially in the large clinical trial space, they were looking at it, and I think ear, neck, and throat maybe but you can go back before they were required to do these large clinical trials and register them on clinicaltrials.gov because they were so big. You got like money trails that you can look at what happened to all these. And so somebody did an analysis like looking way back in the past, basically pre-2000, post-2000, in terms of like the primary endpoints and whether they were whether they met the hypothesis of being significant or not. And you see a rapid drop when they were required to register. And again, like now you're left being like, well, what's going on, right? That's back to your question of what's going on here. These are ones where we do know the answer, right? So we know it's not like disclosure because they're such large that they have records of everything, which is why they're able to do the analysis. The idea is it's a lot of outcome switching, which still goes on, right? But it's harder, it's easier now to hold that accountable because before, you didn't really understand what people were doing. They probably were saying, oh, well, I said that was my primary, but I changed my mind. Now this is my primary, versus in a registration, right? Like you kind of have to stick true to that. And so you're able to tease that apart. But it's hard to tell too, because it's over such a huge time frame that I think you get the classic one, which is, well, maybe just all the low-hanging fruit was taken care of, right? And now it's harder. I don't know if I buy that, but you still hear that a lot that comes through. So that's an older paper that I'm very familiar looking at the same space.
For the record, not that anything's being recorded or anything, can you say a little bit about what this does to discovery science, or to sort of not, again, pre-registration feels very, I'm hypothesizing blah, and I see it or not. Can you give any guidance to people who do want to, or should people still do exploratory things, or how, what can you say about sort of exploratory science in the pre-registration era?
Okay, and you're saying that's just my opinion in terms of like how exploratory research and pre-registration interact in that specific case. Just to clarify, right? That's what you want me to comment on?
Yeah, is this the end of no one should ever do anything exploratory and only do planned things? Or if I wanted to do, gee, I don't know what the hypothesis is, I'm just going to go and see what happens in some big PCA on every brain volume known to humankind in the UK Biobank?
Yeah, I think it's a great question, and I think it's one that every field is going to grapple with for a while. I know NIH is grappling with in animal studies. They still can't figure out if they should do pre-registration, and if so, where. And I think it's really this latter one, which is where does it sit, right? Because it's that huge, right, exploratory all the way up to translational, that huge gap. They don't know what to do with it, if anything. So me personally, I'll give you, this is my opinion, by the way. Anyone else at COS, talk to Brian, he'll give you a different one. I think they both have strong places. I think the key to me is just you need to distinguish the two. So I love exploratory research, and I'm at a stage that I think with a tool like pre-registration and the technology that can support it, I never want to see a paper. I'd love to see a paper where the times you actually say a priori, or I hypothesized, or I tested, that you're actually like you link straight to the pre-registration. Like I want to say those statements now, like to me, the value is only high now if you show me your pre-registration. Otherwise, I don't think I believe it, because if we know there's a way to do it. What that means though is exploratory is great, both before and after pre-registering your study, right? Because you can completely keep exploring, and I think there's nothing wrong with it. So I think for exploratory research, this is actually a good thing, because I think it can actually let us truly explore, completely take it off, say, this is exactly what I'm doing. I don't know if any of it is right, but I am generating a whole bunch of hypotheses. And now if I actually want to test something, I really need to do it the right way. And I think, the question of publication is going to come back up, right? Which is, if we're willing to say, and we do this for large ones, which is, the exploration itself, the data, essentially the data is what we're trying to get at, is so valuable, we should reward it. I think that's a good thing. I think that's exactly what we should be doing, which is reward that massive generation of ideas, massive generation of data for yourself and others. But don't do it at cost that you have to have a neat story that supposedly sums it all up, which is the way we publish now. I see that all the time in the field where I'm coming in. Most of the papers we had look just like that. Huge screens, huge exploration, and then the whole paper gets accepted because they did it in an animal model, and then we can't figure out how to get the same result. Like, I just feel like you robbed exploration at cost of confirmation without actually putting the proper pieces in it. So I don't know if that makes sense, but to me, actually, I think they're valuable both ways. And really, pre-registration can be that perfect tool to say, of everything that I'm exploring, let me make sure I'm investing it the right way so I can actually advance it. But we'll see. It'll take time. But I've done this once or twice in reviewing papers, especially people that I know are aligned, where I'm like, oh, you've got pre-registration. You said the word a priori. Where's your pre-registration? Or just don't use the word. And there's nothing wrong with it. Don't use the word.
Yeah, great. I know we're over time, but thank you for indulging our questions. For the tape, I will remind folks listening that the last few of us who are here, our next webinar is Friday, May 6th. We'll have Eugenio Iglesias from MGH, who works with the Free Surfer team, and I think Fast Surfer, some of the machine learning Free Surfer applications we will hear about next month. So thank you again, Tim. It's great to chat and meet you, and we'll be in touch, going forward.
Awesome. Thank you.